Dr. Gauri Jairath, please, ma'am, start with the introduction.
Sure, sir. Thank you, Dr. Mathi. Good morning, everyone. I welcome you all in the morning session of day 10 of 21 days training program. Today, we have with us Dr. Sandeep Kumar Khurana, who is a renowned scientist in the field of veterinary public health and epidemiology. Thank you very much, sir, for sparing time for us.
Thank you very much.
Now, I would like to take the opportunity to formally introduce him in front of you all. Dr. K Khurana, who is currently working as a principal scientist at ICAR-Central Institute for Research on Buffaloes, Hissar, obtained his master and doctorate degree in veterinary public health and epidemiology from CCS HAU, Hissar. He was awarded gold medal in year 1980 for attaining fourth position in pre-medical examination of Kurukshetra University, Kurukshetra. He was awarded University Merit Scholarship by Kurukshetra University and ICR Merit Scholarship during BVSC and AH. He is also a recipient of ICR JRF for doing MVSc in veterinary public health and epidemiology. He received Professor R.M. Sharma Memorial Award by Association of Microbiologists of India for the best research paper in the field of microbiology in the year 1996. He received Dr. K.S. Nair Memorial Award and Medal for the best research paper in the fields of bacteriology, virology, parasitology in the Indian Veterinary Journal in year 1998. He received Dr. C.M. Singh Salihotra Summon and Citation 2018 by Dr. C.M. Singh Endowment Trust for outstanding work in the field of veterinary public health, epidemiology, and zoonosis. Besides, he had been recipient of several other awards in several activities at college and university level during student life and also during service in various capacities as a scientist. His main area of research including disease of bacterial, mycoplasma and leptospiral origin, including the disease of zoonotic importance with special emphasis on epidemiological aspects, especially serosurveillance with an objective of prevention, control and eradication. His notable contribution in research include development of different ELISAs for diagnosis of mycoplasma equigentilum in equines. He and his team developed molecular diagnostics for Rhodococcus equi infection in foals, including multiplex PCR. He has established seroprevalence of Brucellosis and IBR in different species of livestock in Haryana, which helped him in formulating prevention and control strategies. He has standardized different immunodiagnostics for diagnosis of leptospiral infections in equine and found countrywide prevalence of equine leptospirosis. He has established prevalence and epidemiologist pattern for important diseases of equines of different etiology. He was the main leading scientist who have created a very huge repository of equine serum samples at ICR National Research Center on Equine Hissar. He has worked on glanders since its emergence in 2006. And he was at forefront for detection, surveillance, monitoring, and control of glanders from several states of the country. He is also involved in filing 2 patents on glandar diagnostics, of which one is granted. Presently, he is working on buffalo's disease, particularly on the etiological and management aspects of the buffalo calf mortality. He has more than 100 research and review publications in national and international journals of repute. He has written and edited scores of books and manuals. He is the associate editor of several journals. He is on the regular review panel of several high-impact international journals, besides being lead editor of special issues of research journals. Thank you very much, sir, for giving us time and giving us an opportunity to learn from you. Thank you so much, sir. Over to you, Dr. Sandeep Kumar Khurana, sir.
Thank you very much, Dr. Gauri, for this detailed introduction. I thank organizers, Dr. Nisar and Dr. Radotra, who is also my batchmate, for inviting me for this lecture. I really thank you for giving me this opportunity. I am The topic of the lecture which I am going to deliver is zoonotic diseases transmitted from equines to man. Unlike you can say dog, the common disease rabies everybody knows. The diseases from equines to man are not very much known to everybody, especially the common man. So, but these are important in recent times. Recent times because we don't know which of the animal disease may come as zoonosis to human beings and remain thereafter as a human disease. Like historically, we can say AIDS was not a human disease previously. Similarly, malaria was not a human disease. These were animal diseases, and later these came to humans and became human disease. So the danger of zoonosis is very great, which we cannot foresee, especially in present times when COVID-19 is engulfing the entire world. We don't know its real origin also. Let me take you to your earlier days, simply starting from the definition of zoonosis. Zoonosis is singular and zoonosis is plural. ES is plural. Zoon is animals and nosis are the diseases. So these are the diseases or infections which are shared between animal and man, and this sharing is not unidirectional, not only from animals to men, from man to animals. Some diseases may also be transmitted, and those are also called as zoonotic diseases. Now, such diseases are very less in numbers, which come from man to animals. Zoonosis we can classify broadly on the basis of its etiological agents like viral zoonosis, bacterial zoonosis, then spirochetes like Leptospira, protozoan diseases, and fungal diseases. You can just have a look. I may not read these. Then classification of zoonosis based on transmission. These are classified as direct zoonosis, where the infection goes from one vertebrate host to the susceptible host. Vertebrate host is generally animal, and the susceptible host is man. This goes by direct contact through without the multiplication of mostly without the multiplication of the disease agent, but sometimes this may go some propagative or developmental changes during transmission. The second one is cyclozoonosis, where for the maintenance of the disease agent, two vertebrates are essentially required, like ascariasis and teniasis. And then metazoonosis, where these are vector-borne diseases, we can say vector-borne zoonosis, where one or the other vector is there. These vectors are invertebrate, you can say invertebrate hosts like mosquitoes, cockroaches, midges. The disease is spread through these vectors. Then saprozoonosis, where there is involvement of soil, water, or some inanimate some inanimate objects like pigeon droppings also, where the agent multiplies or is goes some development stages. Then based on whether the major host is of the disease is man or animal, these are based on the basis of reservoir host. This is classified as anthropozoonosis, where the infections are transmitted from to man from lower vertebrate animals like rabies. Then zooanthroponosis, where the infection is transmitted from man to the animal, to the lower vertebrates or animals. The examples are some staphylococcal infections, some skin infections. These may be transmitted from man to animals. Then amphizoonosis, where man and animals have equal stake. means both, that is amphi means both. They have, these diseases are of both man and animals of equal importance. Zoonosis, like other diseases also, there are some vulnerable classes, vulnerable groups. The veterinarians are most, you can say, most susceptible or vulnerable to these. Then paravets, stockmen, animal handlers, animal health persons, pet pet owners. Then some human beings which are at stress, like pregnant women, infants, children's, immunocompromised people, and some human beings which are having some other disease already, and the patients undergoing antibiotic therapy or chemotherapy. This is the common disease triangle which sets up the disease. There is a balance between host and vector and environment, which makes the disease possible. If any of these factors is not conducive, the disease will not occur. And while breaking the cycle, while controlling the diseases, we have to take care. We have to see what is the this transmission cycle or the disease triangle. Recently, many diseases, especially in the past decade, more than 75% of the human disease are zoonotic in nature. And all human diseases, out of all human diseases, 60% of the infections are zoonotic in nature, which we may not know exactly. And as I have already told that this can be classified on the basis of animal, I have already told this about this classification. Many diseases, they come from horses or mules or donkeys to human beings. The most common or most fatal are encephalitic and alpha viruses, then rabies, glanders, anthrax, brucellosis, etc. And in the past, anthrax and glanders, they have been used as main biological weapons during World War I, II, and also later on. And this defense laboratories throughout the world are working on such bioweapons. As already told, that these have been classified based on etiological agents into viral, bacterial, parasitic, and fungal, leptospiral, etc. The common viral pathogens, the most common which are transmitted from equines to human beings are encephalitic alpha viruses, and among them Eastern equine encephalitis, Western equine encephalitis, Venezuelan equine encephalitis, and West Nile viruses are very common. And these have been isolated from horses, human beings, and mosquitoes, birds, rodents, and other animals also. And these are spread through blood-sucking vectors, like mostly the mosquitoes of different species. Now I will tell you something about Eastern equine encephalitis, and this was first isolated from horses in 1933. And this uh, virus is propagated in nature between birds and mosquitoes, and this is perpetuated in nature between birds and the mosquitoes, and man is the accidental host. When this mosquito, they bite human beings, they get the infection, and in especially in the endemic area, and this is very fatal, with case fatality rate as high as 70% in human beings. And horses also may transmit it when this mosquitoes suck the blood of horses, and then again, when again they bite human beings, this disease can spread. And the incubation period is 4 to 10 days. Fever, headache, vomiting, respiratory distress, seizures, and coma are the main symptoms of this EEV infection. And diagnosis, there are many diagnostic tests, and vaccines are also available, but these vaccines are used only in the areas where this disease is common or endemic. This is the transmission cycle of this virus, Eastern Equine Encephalitic virus. The disease is transmitted naturally between birds and mosquitoes. This virus is transmitted, and men and horses, they become accidentally. The human beings get infection from infected horses also through mosquito vectors. The second disease of this group, that is encephalitic alpha viruses, is Western Equine Encephalitic virus. This is also quite similar to Eastern Equine virus, and this was first isolated from brain of a horse in the year 9 and from human in 1938. This is naturally transmitted or naturally propagated between passerine birds. Passerine birds are this common sparrow and some alike birds and mosquito vectors, and some rodents and logomorphs. Logomorphs are of the family of rabbits. They also propagate this virus, and the incubation period is 2 to 7 days, and diagnosis is by ELISA and some other tests, and vaccine for this is also available, and these are used in the areas where this disease is common, not in all areas. And this is the cycle of transmission. This is naturally transmitted between birds and mosquitoes, and also some rodents and logomorphs. And the men and horses are dead-end hosts or accidental hosts of this infection. Then of the same group is Venezuelan equine encephalitic virus, and here this disease is naturally transmitted between rodents and mosquitoes, and this is transmitted to humans when mosquitoes bite humans, and to equids also when they bite the equids, and from this infected equids through vectors, again this can come to human beings. Horses, donkeys, and mules develop sufficient viremia and transmit the infection through mosquitoes. And human beings also can transmit. This disease can be transmitted among human beings also, where they live in very close vicinity and very densely they live. From mosquitoes, they can go from one human being to other also. But this is very, very rare. There are only few reports of this kind. And vaccines are also available. As already told, these are used only in the endemic areas, and some new vaccines, DNA-based vaccines, have also been developed, which are available. This is the transmission cycle. One is between rodents and mosquitoes, and other cycle is when the human beings and equines get bitten by the mosquitoes, and this cycle may also go. The first cycle is called enzootic cycle, and other is epizootic cycle. Means the common cycle, common zoonotic cycle is called enzootic cycle. Then another virus is flavivirus, West Nile virus, and this was first isolated in 1937. And this disease, virus is also propagated naturally between culex mosquitoes and birds, and horses, human beings, and some other mammals. They get accidentally infected through bites of the mosquitoes. And many diagnostic tests of this are available, and this is quite similar to Japanese encephalitis virus also. And there is no suitable therapy at present. Number of vaccines are there, but used in only in the endemic or localized areas. This is the transmission cycle of West Nile virus. This is between birds and mosquitoes, and human beings and horses. They accidentally get the infection. Infection is not naturally transmitted between them. They are accidental or dead end hosts. They don't further spread the infection to other animals. This is about the human cases. Human cases. In human cases, there is involvement of central nervous system, but that is only in less than 1% of the cases. Most of the cases, about 80%, are asymptomatic, and 20% show some mild fever. So this the CNS cases are very less, and out of these less than 1%, only 10% are fatal. So the case fatality rate is 1 in 1,000 or even less. The next zoonotic disease, which is of very great importance, is Japanese encephalitis. Here, thus, the amplifying host are pigs and birds, and this horses get the infection accidentally. And this was first isolated in Japan in 1935, and in India this disease is very common in the northeast area, West Bengal, and up to Bihar. And the for amplification of this virus, pigs play a very important role, and this disease is not there in the areas where pig is not there. Mostly it is not there. In this, you can say in Muslim worlds, in Islamic countries where they don't eat pig, this disease is not there or almost negligible. Through some other animals, it is not in Iraq, Iran, Pakistan. It is not there. So regarding incubation period, its incubation period is 5 to 15 days, and the most of the conditions are asymptomatic, and here there is symptoms of encephalitis like fever, neck rigidity, then paralysis, convulsions, etc. And the mosquito vectors are mostly Culex, and Aedes mosquito also transmit. Some vaccines are available. This is the transmission cycle of this virus. This virus is naturally transmitted between mosquitoes and some, you can say, some birds, like some water birds and pigs. And pigs are the amplifying hosts of this, and horses are the accidental hosts. Then rabies. Rabies is not as common in equines as in dogs, but from a rabid horse or mule, human beings can get this infection, and there are many cases like this which are reported, and these are the equine handlers must be, especially in the hilly areas, they must be very cautious about this, that the horse which is bitten by a dog can get rabid, and then this can further transmit the disease to human beings. Horse handlers, veterinarians, and horse owners are at high risk, and man experiences symptoms like ataxia, listlessness, paralysis also. And horses don't show the typical symptoms as in dog, but nervous symptoms are shown, and there is muscular incoordination in horses. And this can be differentiating from the other neurological diseases. And animals died in between 2 to 14 days after appearance of clinical symptoms. Central nervous system and component and saliva, they are rich in virus. Then Hendra virus is also a zoonotic disease which has been reported in past decade, especially in American continent, and this was first isolated in 1994. And this mainly causes respiratory symptoms and also encephalitic symptoms, and mortality may exceed 70% even. And the horses acquire infection from flying foxes. So flying foxes are like you can say like bats. Clinical signs in horses initially include fever, anorexia, followed by respiratory signs, and humans get infection through direct contact with secretions of horses. Fruit bats also play a role in its transmission. And veterinarians, while examining the oral cavity of the horses, should be very cautious, because this saliva is a rich source of this virus. Many diagnostic tests are available. You can just have a look here. These are some of the viral diseases. The list is not, you can say, exhaustive. It is not complete. There are many other viral diseases also, but these were the important viral diseases which I had mentioned. Now we will talk about the bacterial diseases which are transmitted from horses. There are a number of diseases like glanders, then Rhodococcus equi infection, Brucellosis, Anthrax, Streptococcus equi subspecies Zooepidemicus. This is different from Streptococcus equi subspecies equi, which is cause of strangles. Strangles is not zoonotic, but this is zoonotic. Then Clostridium difficile infection, Somnosis, Mrsa, and Tuberculosis. And there are some others also. I will describe in brief one by one. This is again a pictorial representation of diseases which can be transmitted from equine. Equines I will mention at some other points also. You can just have a look on this. The glanders is the very important disease which is transmitted from horses or mules or donkeys to human beings. And all of you must be knowing something about this disease. And this glanders agent is the bacteria is Burkholderia melii. Previously, it was called Pseudomonas melii, and this has been reclassified several times, and presently it is called Burkholderia melii. And this disease has been used, or this disease agent has been used as a bioterrorist agent, because only a small amount, a very little amount of this bacteria can spread disease among many in large number of horses and human beings. And common modes of its transmission are inhalation and ingestion. The disease occurs in chronic form in horses, whereas in mules and donkeys, the disease is in acute form, and the acute form involves pulmonary, cutaneous, and nasal type of symptoms, and these which are characterized by fever, cough, nasal discharges, ulcers, on mucosa and throughout the body, and nodules on skin, and finally this leads to death. And this is a notifiable disease in India, and treatment is not recommended in India. Some countries they recommended treatment, and is in some countries this disease is not a notifiable disease. Human glanders, we have our own. Our team had our own experience at National Research Center on Equines. This disease has resurfaced in the year 2006 after about 2 decades of its dormancy, and we had seen many cases in from Pune and Panchgani initially 2000, after a gap of about 2 decades, and since then we have reported or NRC has reported several cases. Each year more than 100 cases are still coming, but we have not come across even a single human case during this means more than 500 animals have been sacrificed, which were found positive of glanders, but no human case was seen. So this disease is not common now. This is my own analysis, that this disease was very much zoonotic, and large number of soldiers and common men were infected during World War I and II, and thousands of humans had died during that time. That time, I think the conditions were very different. The soldiers were living in very dense barracks, and they were in touch with horses which were wounded and having glanders. So this condition is not there. Perhaps that is the reason that we are not coming across human cases at present. Though there are many cases among horses, mules, and donkeys. And this has been used as a biological weapon during different times, especially during World War I and II. The internationally recognized test for this is complement fixation test, and other tests are indirect heme agglutination test, ELISA, and a Malin test is also used, which is allergic test. But since it is difficult and cumbersome and time consuming, CFT is used as a test for the international trade. And our team had also developed an ELISA which is being used at present by many labs. And treatment is not recommended, and also no vaccine is available for animals or human beings for this. And whenever a suspected case of glanders is there, which is dead, the carcass is buried deep or incinerated. These are the few cases. You can just have a look, which we had seen during our visits, or which we had seen at different places. You can see. You can just have a look of the lesions of glanders, in horses. And all these ponies, these are ponies, small horses. All these were sacrificed later on. Then other important zoonotic infection is Rhodococcus equi infection, which primarily causes pneumonia in foals and also pneumonia in human beings. And this disease is transmitted from horses to human beings very commonly. And the main symptoms are respiratory symptoms, or main disease is pulmonary, but extra pulmonary complications like enteritis, arthritis, and abscesses in abdomen is also there. And this was first this organism or this bacteria was first recovered from lung of a foal, and that at that time this was known as Corynebacterium equi, and later this was reclassified as Rhodococcus equi. And this is very important in AIDS patients. In AIDS patients and also tuberculosis patients, this infection occurs concurrently, almost in one-third of cases. This is very, very common, and this disease requires a very long treatment, just like tuberculosis treatment, and similar type of treatment is required because this organism also requires resides inside the cells, and antibiotics, uh, most of the antibiotics are not able to reach there, and this requires the same regimen as tuberculosis, like rifampicin and erythromycin combination. Here the virulence associated protein is associated in foals, whereas virulence associated protein B is associated in human cases and also in pigs. And this Rhodococcus equi infection is found in many other animals also, like camel also, and some also in some wild animals also. And human acquire this infection mainly through inhalation of the dust harboring bacteria from domestic animals, including equines. Uh, we in our lab have developed ELISA and PCR assay, which is being used internationally in various part of the world. And rifampicin, along with some macrolides like azithromycin or erythromycin, are the drug of choice. Proper management, sanitation is very important to control or prevent this disease. Here, this we can say a foal lying, having respiratory symptoms, and the other picture is of brucellosis, whereas this is showing fistulous with this. Now I'm talking about brucellosis. Brucellosis in equines is mainly caused by Brucella abortus, like in like the Brucella which causes disease in cattle or buffaloes, but the symptoms or the manifestation is quite different. The brucellosis in horses primarily causes fistulous withers or pole level, and rarely late abortion in case of mares. And horizontal transfer of Brucella species to horses from cattle and pigs is also reported. As you know, this is also a type of occupational disease which is affecting slaughterhouse workers, butchers, and veterinarians, and this causes undulant fever in human beings. We have seen many cases of brucellosis in veterinarians, and well, especially when they are handling the abortion cases. An incubation period varies from 1 to 3 weeks, and there is, as you might be knowing, there is undulant fever. Means there is a fever for 1 or 2 days, then there is no fever, then again there is fever. Headache is there, weakness is there, body pain is there, and especially night sweating during night time, profuse sweating, then cough, chest pain, irritation, insomnia, depression are also there. And if there is no treatment, or if it goes undiagnosed, the other complications occur, and this spreads to other organs and may lead to death if it is not diagnosed, and proper treatment is also not given. Many tests for its diagnosis are available. Most common is Rose Bengal plate test, and other is standard tube agglutination test. Then several new tests, PCR and real-time PCR, also available. And the treatment in human goes for many months, 6 months or more, and a combination of doxycycline, erythromycin is most common, and rifampicin is also used. Then anthrax. Anthrax is caused by uh, the bacteria Bacillus anthracis, and this affects many species. And horses are one of them. And again, horses get infection by grazing of the areas contaminated by the anthrax bacillus, and humans coming in contact with such horses or from infected horse meat, they can get the infection. And incubation period is typically 3 to 7 days. Symptoms appear very rapidly, which include high fever, chills, colic, anorexia, labored breathing, seizures. Bloody diarrhea is also seen. This is seen in all other animals also, and death occurs very soon, within 2 or 3 days. But horses are less susceptible than ruminants, less susceptible than cattle and buffaloes, and the disease could be prolonged. This may prolong for a few weeks also. And human beings get infection from contaminated horses and accessories, and often show three forms of, that is, cutaneous, gastrointestinal, and inhalational form. And this bacteria has been used as a bioterrorist agent during World War One and Two, like glanders also. And recently, not very recently, in the year 2006, this has also been used against USA. This the anthrax bacillus powder was sent through postal means to some senators of USA from Iraq or some other countries uh, in 2006. So this has been used potentially as a bioterrorist agent also, and this waterways were also polluted with this, and during World War One or and Two, and this has led to great devastation and many deaths during that time. And after that also this has been used by many countries, and this along with glanders and brucellosis are being researched as bioweapons by many countries. Then again, there are some other bacteria like Streptococcus equi subspecies Zoonotic, which is zoonotic to human beings, and this is an opportunistic pathogen of horse, but it also infects cattle, sheep, goat, pig, and dog also. And this is very similar to Streptococcus equi subspecies equi bacteria, which causes strangles, a common disease of horses. But strangles is not a zoonotic disease, but this is a zoonotic disease. So, and various type of detection methods are available for this. Then again, Clostridium difficile infection is also common, which causes colitis and horses and humans, and transmittable from horses to humans. And this may lead to death in horses. The infection in man varies from mild to severe, leading to intestinal perforation and death also. And here, this sanitational and public health measures are important to control and prevent this infection. Then again, very common, one of the most common diseases is Salmonellosis. There are several serotypes of Salmonella bacteria, and this is so widespread that it is found from elephant to fishes to reptiles to insects. Everywhere Salmonella is very common, and this organism becomes fatal also many a times. And this leads to diarrhea, fever, which may lead to acute toxic enterocolitis or septicemia in horses and humans. There is one multiple drug-resistant Salmonella, Salmonella typhimurium DT1 104, which is having a high zoonotic potential. Initially, it was isolated from cattle, and later it was found in sheep, pigs, poultry, and horse. So horse is also important source for this. And fecal oral route is the most common route for this zoonotic transmission to human beings. Immunocompromised persons are more susceptible or vulnerable, and higher mortality rate and resistance to antibiotics adds to its zoonotic potential. As you know, for salmonellosis, proper cooking of food and proper hygienic measures of in the stables are important to spreads to prevent the spread of this zoonotic transmission of salmonellosis. Then MRSA, which is in news for many, you can say for many years now. The horses can harbor this organism. This is methicillin-resistant Staphylococcus aureus, MRSA, and about 10% of the healthy horses harbor MRSA. So this is very dangerous. Even healthy asymptomatic horses harbor this, and handlers can get this this infection. And since there is an international trade of horses, which is very common for different support purposes, they also go. So while this international movement of horses, this MRSA can also spread, and veterinarians and animal handlers are most susceptible or vulnerable persons in this. Then again, tuberculosis. Tuberculosis is also found in horses along with other animals and human beings. This is assuming a great importance in the present days because multiple drug-resistant TB strains are there, where the common antibiotics or common drug therapy which was used against tuberculosis is totally ineffective. So there is XD TB, that is extremely drug-resistant TB, then total drug-resistant TB, TDR TB, where no drug is acting, and if some patient gets this infection, there is no escape. He has to die. So this is the situation nowadays due to spread of this MDR strains, due to, you can say, overuse of antibiotics, we can say. And incidences of TB are rare but not uncommon in horses. And this is in horses is caused by Mycobacterium tuberculosis and bovis also. And horses residing in close proximity with infected cattle also acquire this infection. Then there are few parasitic pathogens which are important in horses and can spread infection to human beings. And most important among them are Cryptosporidial Sporidiosis, that is caused by Cryptosporidia parvum most commonly. This is a protozoal disease. And horses develop asymptomatic Cryptosporidias, and human get infection from them. And while handling the diarrheic animals, care should be taken that this causes diarrhea in foals and horses, and human beings can get infection from them. And this disease is of severe nature in human beings than horses. Then GRDSs could have many sources, many animal sources, and horses are also one of them, a very important source of Giardia intestinalis. And there is asymptomatic shedding of Giardia in 25% of adult horses, and in 71% in foals. So this has tremendous potential for spread to human beings. And detection methods of fecal cyst detection and ELISA are also there. Then apart from this bacterial, viral and parasitic, there are some others also. Leptospira is a spirochete which is a bit different from the true bacteria. Then some fungal infections are also there, that like trichophyton, and symptoms in horse are mild or subclinical form. These are dermatophytosis is like amphizoonosis, which is shared equally between human and horses, or human and other animals. Then leptospira is also very important. This may cause jaundice, fever, muscular pain, vomiting, uveitis in human beings, and also this may cause abortion in human beings also. Forgot to mention here. Zoonoses are quite different than the other diseases because these are the diseases about which the veterinarians are not fully aware, and medical persons have a little knowledge about this. So such diseases they fall under no man's land. But as no one is ready to take the responsibility of zoonotic disease, neither the veterinarians nor the medical persons. So these are the, you can say, neglected diseases. There are budgets for many human diseases like vector-borne diseases like malaria, now COVID-19, and animal diseases like FMD, HS, etc. But zoonotic diseases, there is no large separate budget for zoonotic diseases, which which are assuming great importance nowadays because of this One Health approach, and they can spread from human to animals or animal to human, and can cause uh, uh, a lot of lot of damage in terms of finances and human and animal deaths also. There is weak surveillance system of the zoonotic diseases. There is lack of scientific surveillance. Zoonoses are often made undiagnosed. Whenever somebody, I just want to give an example. uh, if some veterinarian suffers from brucellosis and he is not knowing that it is brucellosis, and he goes to a medical person for treatment, he can never diagnose it unless he himself diagnoses it by knowing the history, that whether he has handled some abortion cases like that. So this is a great problem. Again, which I have told, that lack of awareness on no man's land appears to be a nobody's responsibility. In human animal episodes, neither human nor the veterinary health system have the capability or the, you can say, will to do to work on this such type of diseases. And there is lack of intersectoral approach, which is very much required between human, animal, and animal scientists, workers, veterinarians, and public health workers. How we can reduce the risk of zoonosis? I'm not talking about equines alone now. That is early diagnosis, like other diseases also. This is important, but here it is more important because we cannot diagnose these diseases because we don't know the history, whether human being has come in contact, whether this disease being can get from animals. So diagnosis is delayed or there is misdiagnosis because of this reason. Then personal hygiene can reduce the risk of zoonosis and also all other diseases also. Then identification of potential fomites or vectors can reduce. Protective clothing for laboratory workers, and use of latest therapeutics and vaccines. And these equines are these ponies and the mules are used for production of antivenom and antitoxins like tetanus. So while producing these, the horses should be screened for zoonotic diseases also, apart from the common equine diseases. Other diseases must also be checked, and proper screening should be done so that these such diseases do not spread from horses. What we can do in future to reduce the risk of zoonosis, and especially the zoonosis from horses. We have to strengthen the risk assessment, enhancing our laboratory capability and capacity for diagnosis, and proper monitoring and treatment is required. As I have just now told, testing of antivenom and antitoxin produced from horse for various disease pathogens can prevent diseases. And we have to take precautions for breaking the transmission cycle of diseases, especially in viral diseases, which I have mentioned earlier, that encephalitis arboviruses. Some vector is there, so which is essential for transmitting the disease. So if we can control or kill this vector, that is mosquito in this case, so the cycle is broken, and thus we can prevent. So in that case, in such diseases, the approach should be to control the vector. So we should know the transmission cycle exactly, the season in which these mosquitoes or any other vector is comes into breeding, so we can control it in time. So such scientific knowledge is essential to control such diseases. Then appropriate early diagnosis, high personal hygiene precautions while handling animals for mites and various samples, and medical professionals, veterinarians, and public health personnel must coordinate to control this. Then approaches to control one type of zoonosis differs from other type of zoonosis. For example, in Cyclozonosis, where there are two vertebrates essentially required for transmission of cycle, the approach is different. Here, we have to control the agent. Like in Ascariasis, we have to work against the ascarid or ascarid parasite. So in Cyclozoonosis, there are different approaches. So depending on the type of zoonosis, we have to act. And for metazoonosis, in fact, this is directed against the vector or the vertebrate host or the both. And there is requirement of a large amount of funds and coordination between national and international agencies, a better coordination in future, and better funding, implementation of good management practices, biosecurity, quarantine, etc. is required. Then optimum utilization of the new diagnostic or advanced diagnosis and surveillance techniques, this is required. And not to mention that One Health approach, which is getting importance nowadays, this should be taken into consideration to control the disease in the environment, both for human and animals. Thank you.
Thank you very much, sir.
Thanks.
You may now unshare, and now we will discussing about the question. We will take the questions written by the participants. So one person has raised the question that for rabies, equines are dead and host, isn't it? What is the chance of transmitting rabies from horses?
Actually, this was not audible. Please repeat your question.
So you may also see in the chat box. The question is, equines are dead and host.
Yeah. Yes, many diseases.
What is the chance of transmitting rabies from horses?
Yes, there is a chance of transmitting rabies from horses, because it is in the saliva of horses also in later stages, and and they can bite the handlers, or the handlers come in contact with saliva. They can, but this is
পিটবোৰ যেতিয়া ক্লিন কৰে ন saliva-টো পৰি থাকে ন এটোৰ পৰাও মানে
so there are chances, but it is definitely less than dogs or cattle.
Okay, sir. The next question is, who discovers brucellosis disease? You may also, the participant may also Google it. Who discovered brucellosis disease?
I just don't remember. I just
Sir, I have Googled it. Sir, I have Googled it. It is discovered by Sir David Bruce, the
yeah.
military physician who discovered the etiologic agent Brucella melitensis.
Okay, this type of you can say this type of things you can see you know.
Which Quran? No, he meant to say who diagnoses. কৈছে ন মেডিকেল ডক্টৰে ডায়াগনচিছ কৰিবই নোৱাৰে বুলি কৈছে ন সেইটো ক'ব নালাগে আজিকালি কৰে ব্ৰুচেলা পুৰা কৰে হেণ্ডলাৰ্ছ বুলি ক'লে মানে সেইটো চাইডত চাই নেক্সট
Lima Mora, ma'am, you are not audible. Please raise your query clearly. Lima Mora, ma'am, is there any query at your end?
No, actually I wanted to know regarding that diagnosis part of brucellosis. Like sir said that it's very difficult to diagnose the brucellosis once it occurs. So like from yes sir.
No.
In case of human.
No, it is not difficult. Same.
Okay.
Yes, this is standard tube agglutination test. This is used for human also.
Okay.
Okay, this is not very difficult. What I said, this is difficult. The diagnosis is difficult by the test is there, but human beings, they don't know that this is this disease, so they don't go to the proper person. If they suspect it is brucellosis, they will go for its testing, but they don't know that this is some diseases like this is there.
Okay, thank you, sir.
Thank you, ma'am.
I have many I had one experience myself. I was not working on this brucellosis earlier. So one of my friends, close friend, had symptoms of night sweating and fever, underlying type of fever. So he just he was residing near my house, and he telephoned me that I want to go to doctor and I am having fever. I was not knowing the details of his disease. I went to a leading physician here at Hissar with him, and in the way he told that he is having symptoms like that. Earlier I was not knowing that, and when in the way I told that it could be brucellosis, and I told the physician also that I suspect it is brucellosis, and he laughed at me that this disease I had studied during MBBS 30 years ago. I have not seen any case like this nowadays. So this was when I diagnosed, when I tested in my lab and also at HU. This was there was a high titer of brucellosis. So this is the case that medical doctors are not they are not aware of this. So in this aspect, I told that this is diagnosis is difficult. Tests are there, tests are easy, but they don't know the history or other things. Am I audible?
Yes, sir, you are audible.
Okay.
Okay. Sir, the next question is vaccination is necessary after vaccination second time dog bite in human?
You're talking about which disease?
So dog bite. If there is a dog bite in a human and he is vaccinated, and
Yes.
after vaccination,
Yes, it is
second time
necessary.
dog bite.
Yeah. Second time is also it is required.
So within 1 year also it will be required?
Yes, it is better.
Okay.
Because the present state of vaccines, yes, you can say that it is thought that it will prevent, but present state, of all vaccines, whether it is FMD or COVID vaccine or any, we should not go for this because it is a question of human life.
So the next question is, please suggest some preventive vaccines for equine veterinarians.
I don't suggest any, because we have to see this how common it is, how common it is. So it is not very common in endemic areas. uh, means in endemic areas, not only there are means there are only few vets which are working solely on horses. So we can have this prevention from tetanus only, not many.
Okay, thank you, sir. So the next question is, which organ is affected when brucellosis infection occur in male and name of the condition?
Actually, in male, the this orchitis is also very common. Orchitis in later stages.
Okay, sir. Next is, sir, please suggest some vaccination schedule for equines.
Vaccination schedule as such, being a public health person, as such, I cannot just tell, but uh, we are not going for any vaccination schedule commonly. We are going for this tetanus prevention only.
Very well, sir. I guess no other question is there. Is there any question, dear participants? Okay, so we were having one question more. How to prevent Nipah virus infection?
Actually, Nepa virus is, you can say, it has appeared in India in some states, Kerala, etc. And this bats, or this fruit bat, it is coming. So main thing is that we have to means guard against these bats only, or preventive because it is not very common, so preventive vaccine also is not suggested. So whenever this in some area it comes, we have to go for mass surveillance, etc., zero surveillance.
Okay, sir. Thank you so much, sir. There is no other question. Thank you so much, sir, for sparing time.
Thank you.
Thanks.
Thanks, organizers.
Sir, there's one question. I'm sorry again. There's one more question. Sir, if 1 month old puppy bite a child
Yeah.
having an age around 5 years, is
Okay.
vaccination or treatment required?
Actually, theoretically, if you say that puppy of 1 month will not be having rabies infectivity, but we should not take risk. I will again say that vaccine is required.
Okay, sir.
Is it okay? We cannot
Yes,
take
sir.
risk.
Thank you, sir. Is there any other question, dear participants? Okay, so we are done with the question session. Thank you so much. Okay, we are again having one. When area is affected with flood, then which method is suitable for carcass disposal of anthrax animal?
Burying deep.
If area is affected with flood,
In areas, okay, I didn't listen that. Then incineration. In areas affected with flood also, all human beings are living somewhere. The ground is there in areas affected with flood also. Then incineration is also difficult. So this burying deep is the only solution.
okay, sir.
So you're talking of rarest of the rare situations.
Yes, sir.
Is it okay?
Sure, sir.
Main thing is that this should not come in contact with uh, any human being or any other animal.
Shall we close the query session or is there any other?
Sorry, I think there is no question now.
Yes, sir.
Thank you, Doctor.
Thank you so much, sir. Thank you
Thank
again
you very much.
for the wonderful presentation. Thank you, sir. You may leave now,
Thanks,
sir.
Dr.
Thank you, sir. Thanks a
Thank
lot.
you. Thank you. Thank you very much.
So, dear participants, in the next session, we will be having Dr. Krishnappa, and he will be presenting on estrus synchronization and artificial insemination in sheep, facts and challenges. So, meet you at 2:30. So, thank you a lot. Stay safe. Thanks a lot.